It's my distinct pleasure to introduce Karin Musier Forsyth as a keynote speaker tonight. She had a distinguished, I don't want to say long, distinguished career. And, you know, I'm going to have a few slides just simply to make sure that I don't forget because I had too much cheese earlier. Karin got her PhD from Cornell University in 1989. So I'm telling you, I'm slurring the word. 1989, is that correct? 1899 will be too long. Yeah. And then did postdoc with Paul Schimmel at MIT, after which she took her first independent position at the University of Minnesota, raised through the ranks, later was named a Merck chemistry professor, and then distinguished McKnight professor, university professor. In 2007, she moved to Ohio State University, where she is now a professor of chemistry and biochemistry and an Ohio eminent scholar in biological micromedical structures. She's also the director of Ohio Center for RNA Biology. So on top of this, she's received many honors and awards. So I could basically read this Campbell Dreyfus Teacher Scholar Award in '96, Pfizer Award in enzyme chemistry in 2003, and she was elected a fellow of AAAS, and she's a fellow of American Society for Biochemistry and Molecular Biology. She's also an associate editor of Journal of Biological Chemistry and on the editorial board of several other journals. She published impressive number of papers, 187 and 33 reviews, commentaries, and tributes, book chapters, 1 patent. She maintained impressively continuous NIH support for more than 30 years and served as NIH HVCD study section member and ad hoc reviewer for a bunch of other study sections. She's serious about mentoring. She just, I don't know how she does this, but mentored 42 postdocs, 53 PhD students, 14 master students, and countless number of undergraduates. So she's also a regular attender of Cold Spring Harbor meeting. We appreciate their support. And more importantly, as part of her hobby, I was told she's a great piano player. So, I mean, maybe we should give her a choice whether she could. We happen to have a grand piano here, so it's up to you what you're going to do. Without further ado, I'll give you Karin.
Well, thank you so much for that wonderful introduction, Greg, and thank you and Florence for inviting me. This is a terrific honor, and I can't even believe it, knowing the list of people who have come before me to give this keynote lecture. But what I really don't appreciate is having to come after a Nobel laureate, the founder of the field, and then coming after me, being squeezed between Michael Emmerman, who is another father of the field. And so I'm going to do my best here, and hopefully you'll find some of what I have to say today of interest. And so my lab is really divided between amino-acyl tRNA synthetases and retroviruses, and the connecting bridge between them is tRNA molecules. And before I start, I just want to point out this structure that I put up here, which is a structure solved by Swapnil Davakar in the Yangzhang lab in collaboration with my lab, where we recently published the fully modified human tRNA lysine 3 tRNA, which has 14 post-transcriptional modifications bound to human lysine tRNA synthetase. So I'm going to tell you stories today which involve these molecules, and if you're interested in the structure, which I don't have time to talk about, please check it out. There's a lot of interesting things here, especially related to modifications. So before I begin, though, I want to remind you of a famous exchange, and I think that we can all relate a little bit to Frodo, or maybe a lot to Frodo, who has a very heavy heart and burden at this time. But we have to remember the words of Gandalf, that we have to decide what we can do at this time, that is given us. And so whether you're inspired by words of country music or the more inspiring words of Maya Angelou, I just want to kind of reiterate a little bit about what David Baltimore said, and that I think it's so important at this time that we come together at these conferences, we share science, we continue to collaborate, and we continue to put science out there and do the best science we can, given the circumstances. And I know that a lot of the students that are asking for the retro cards are asking for advice, and so from the PIs. And so I have 3 pieces of advice that I would like to share today. One is that you should continue to pursue your passions, because that's really what's going to make you happy. You should also figure out what makes you unique, because that will allow you to make unique contributions to your field and make you successful. And then finally, you should remember your journey, because we all have journeys, personal journeys, scientific journeys, and when we go back and think about the past, we think about who inspired us, who mentored us, and who really gave us good advice in the past. That can really help us moving forward. So today, I'm excited to tell you about my journey in science, and this began actually at Eckerd College, a small liberal arts college in St. Petersburg, Florida, where I had really supportive mentors, and I think for sure I would not be where I am today if it weren't for that. This is Gordon Hamas, who is my PhD advisor, also a wonderful mentor. And at Cornell, I learned about mechanistic biochemistry, enzyme kinetics, and some techniques that I learned and became pretty expert at are cross-linking and FRET. And you're going to hear a little bit more about FRET today. My postdoc was with Paul Schimmel. And Paul Schimmel continues to be one of my most valuable mentors and colleagues and friends. In Paul's lab, I really learned about the enzymes that I'm the most excited about, not polymerases, but aminoacyl tRNA synthetases. Learned about the idea of tRNA identity, which I'll tell you a little bit about. Began to learn skills in molecular biology and chemical RNA synthesis. So back then, you couldn't actually buy a chemically synthesized RNA from IDT. You had to chemically make it in the lab, and the chemistry for doing that and for optimizing that was being carried out at MIT while I was a postdoc, and so I was able to collaborate with that group and do some really unique things with chemically synthesized RNAs. I also began to read about tRNAs as primers in retroviruses. I didn't work about it at the time, but I started reading the literature and thought that was super interesting and something that I might want to pursue. So Paul Schimmel I've heard him say this many times, that the greatest discovery of the 20th century was the cracking of the genetic code. And what I have to say at this meeting was that's the greatest discovery prior to 1970, of course. So here are some famous players in the field. And back in 1955, it was actually Crick who first proposed that there must be an adapter between the codons, the nucleic acid sequence information, and the amino acid sequence information of proteins. And he didn't know what it was, but a couple of years later, I'm giving you a little bit of milestones in tRNA research here. After the adapter hypothesis was proposed, Zemenchik, Nick, and Hoagland actually showed that sRNAs, which stood for soluble RNAs, were the adapter molecules. And they also first showed that the amino acylation process, charging the adapters with amino acid. In 1965, the first full tRNA sequence was sequenced by Hawley at Cornell. And then I'm going to get now into a little bit of history of retroviruses and tRNAs. So, Steve Goff can maybe correct me if I'm wrong, but I think the first host tRNAs packaged within retroviral particles was really clearly showed by Jim Dahlberg in about 1973. The 3D structure of tRNA was solved about the same time, and then again Dahlberg in 75 showed that tRNA trip served as the primer in RSV. And this is an amazing paper where he actually elucidated the sequence of the tRNA, including all of the modifications. So encourage you to check that out. And then in 1985, the HIV-1 sequence suggested that the tRNA that primed in HIV was human tRNA lysine. And so this is where I kind of come into the picture here. In 1988, it was 2 groups published some landmark studies showing that the identity element, and I'll tell you what that is in a minute, of tRNA alanine was a very specific and simple G3U70 base pair in the acceptor stem of that tRNA. And the idea of a second genetic code was proposed. And so what do I mean by tRNA identity? So tRNA identity is the specific recognition rules used by aminoacyl tRNA synthetases to select and amino isolate the correct tRNA with the correct amino acid. And so I'm just going to summarize here for 8 different tRNAs. These, what we now know, are the primary identity elements. And this does vary a little bit across species. But basically, in alanine, it's universally conserved. The G3U70 is critical for telling alanine tRNA synthetase to charge this tRNA with alanine. And the yellow circles indicate secondary elements. So it's not quite as simple, and it's not quite a true second genetic code, but there is a type of a code. And so each synthetase uses certain elements on the tRNA, mostly clustered in the acceptor stem and the anticodon, with some exceptions, to recognize their tRNA substrates. So what unique questions could I address with the skills I had, when I was starting my own lab at the University of Minnesota in 1992? And so what I was thinking at the time was that interesting questions include, how are specific tRNAs selected for packaging into retroviruses? There seemed to be correspondence between the type of tRNAs used and the different retroviruses. So is there a code similar to the one that I was involved in uncovering for, aminoacyl tRNA synthetases for recognition of these tRNAs by retroviruses? In addition, how are tRNAs unwound? These are, these rock solid, little RNAs in the cell, and how in the world are they unwound and annealed to the primer binding site when most of the interactions involved in that are involved in base pairing? So I clearly wanted to try to get into the retrovirus field, and so I was given some advice that I should go to this meeting. And so in 1998, I started attending Cold Spring Harbor, and actually here is where I really learned my retrovirology from many of you in the audience. And I actually had never taken a viruses course in my life. So I had to learn all of that here and other places, obviously, as well. But I thought it'd be fun to see what you guys looked like when I first met you in 1998. So thanks to the Cold Spring Harbor archives, I was able to come up with these photos. And I can easily pretty much recognize, everybody. Of course, Henry Levin looks exactly the same, and so on. But I had a little trouble with this person here. And so I actually had to zoom in to look at his name tag, and that is Chris Aiken. So the mustache really threw me off on that one. And the hair. Okay, so here are some more. And I really picked photos of people that I remember early on having great conversations with and really having had an influence on my education in retroviruses here. And many wonderful women role models in the field as well, and more pictures are shown here. I love this picture. This is actually maybe a little before 1998 because I couldn't find one in the 1998 archives. But wonderful people. Johnson Mack here, who's back at the meeting from Australia this year, 1998. And then Akiro. So Akiro still looks really young. I actually told him that today. But he looks about 19 here, doesn't he? He's like... Okay, so it's only fair that I show you what I looked like in 1998. So here I am. We were able to have the wine on the lawn that year, and talking to, again, some people that I met for the first time, and really having a great time learning about retroviruses. I also wanted to show a couple of other photos here. So in 2015, both Larry Kleiman and Judith Levin retired, and I think it was Kathy that organized the meeting, Kathy Boris-Lowry, that organized the meeting that year, and she honored them, or just had some fun and had us all sign a t-shirt for them. And these are two special colleagues in the field that I collaborated with over the years, and of course Judith passed away a year ago or so, and we now honor her with this prize that was awarded yesterday. About a year after this, this was the photo of after Alan's keynote talk. Okay, so back to some science. So the first question we really addressed in my lab in terms of retroviruses is how are the tRNAs unwound and annealed to the primer binding site? And so the main player in that is the HIV nucleocapsid protein. And so this protein has 55 residues. The joke in the field is that there's a function for every residue, which is just about true, and has some really interesting characteristics. About the time we were starting this work, Mike Summers had published a lot of interesting structures, so we started to understand about how NC bound G-rich sequences and Yeah, and then, but the big question was, what is the mechanism of NC's ATP-independent chaperone activity, which ended up being key to tRNA annealing? And let me show you what I mean by NC's chaperone activity. So if you take an in vitro transcribed tRNA and a bit of the primer binding site region of the genome, and you incubate those together under physiological conditions in the absence of NC, we estimated that the half-time of this reaction would be 24 days, right? So this is not going to happen spontaneously. Yeah, that's a little bit of a joke, but we got a little bit of band on the gel after about 3 days, and this is what we came up with. But if you add nucleocapsid protein at the ratio that's actually found in the virus, under physiological conditions, the half-time of that reaction goes to 14 seconds. And so this is an incredibly robust chaperone, accelerating this reaction 10 to the 5-fold. So how does this work? Well, again, at the time we were starting this work, there was some controversy in the literature, and it wasn't at all clear. Some people published that it was that you bind NC and it unwinds RNA. Others said it's a single-stranded binding protein. So wasn't really clear. And here I came back to my roots, my FRET roots, like understanding how to specifically label molecules and then measure distance changes by FRET. So my first PhD student in the lab actually took on this problem, doubly labeled the tRNA. This took about 6 months to really perfect and to get pure material to do this experiment. And this worked, was successful and ended up being my very first talk at Cold Spring Harbor Lab. And so I'm going to show you the results here. And so what we saw was that this is donor-only labeled tRNA, the emission signal of that. When we had the second dye on there, what we saw was a energy transfer, which is indicated by the decrease in the donor fluorescence and the increase in the acceptor absorption. And we could calculate the distance between these dyes to be 33 angstroms. Okay, so what happened when we added the nucleocapsid protein? We expected complete unwinding of this tRNA. Absolutely nothing. So this is the spectrum in the absence of NC, and this is the spectrum in the presence of NC. So this was surprising and unexpected, and we were a little disappointed. We thought we had a project, maybe we don't. But then we thought, well, what if we add the complementary primer binding site? What would happen? And so then we saw, under the exact same conditions, if you have the template there that the tRNA has to anneal to, you get, again, a complete recovery of the donor fluorescence and a complete decrease in the energy transfer. And so this was consistent with annealing of the tRNA primer. And so what this data showed us is that the tRNA annealing really occurs in synchrony with the annealing, or unwinding, sorry, occurs in synchrony with the annealing to the HIV genome. And the other, and we went on for many years later to study NC's chaperone activity. I'm not going to go through all my subsequent talks at Cold Spring Harbor, but just to show, just to give you some of the highlights that NC as a chaperone is really a weak duplex destabilizer. It's a really critical part of its activity, but it really has negligible effects on RNAs or DNAs that are too stable to start with. However, it has very strong nucleic acid aggregation activity, and that really increases the probability of nucleation of two species together, and that is the major force in annealing, or the major part of the activity that's important for annealing, for example, the tRNA to the primer binding site. This activity is mainly mediated by the positive charges, and then this activity is mediated by the zinc finger structures. And so, again, over the years, we published and did a lot of work with NC, and I just want to highlight some of the people that are really, continue to be and work, really key collaborators in this work. So Judith Levin, Yulia Rosina, who's still a senior scientist in my lab, Mark Williams, a lot of single molecule studies, DNA stretching studies, and we continue to collaborate with him. Alan Ryan was important in this work, the late Paul Barbaro, did a lot of single molecule studies with him, and then Rob Gorlick was instrumental in supplying the world really with highly purified nucleocapsid protein in lots of different variants. In 2005 and 2010, Judith and I, together with collaborators, wrote a couple of reviews on chaperone activity. I think this continues to be one of my most highly cited papers ever. And then just a couple years ago, we published this molecular genetics compilation of papers or reviews, and or organized that, I should say, and the publications were by almost everybody who published in this article is in the audience today. So I really encourage you for look up this very exciting, I think, compilation of reviews. Okay, so on to the second question. This one ended up being more challenging, I think. How are specific tRNAs selected for packaging into retroviruses? Again, this idea that maybe there's some sort of a specific recognition process or code, that's involved in this process. So this work was really inspired by Larry Kleiman, who was a longtime participant of this field and again retired in 2015. And Johnson Mack is on this paper, as you'll notice. And so I was attending a meeting where I really heard Larry talk about this for the first time, I think in about 1995 or so. And what he showed was that these spots correspond to specific tRNAs that were packaged into HIV particles. And quantitatively, he showed that about 60% of the tRNAs that you find in HIV are tRNA lysine species, and here's the breakdown within that of the tRNA lysine isoacceptors. Lysine 1 and 2 is a little bit confusing. This is a single isoacceptor. It has the same anticodon. That's what an isoacceptor is. 1 and 2 differ by a single base pair in the anticodon stem. tRNA lysine 3 is a different isoacceptor, different anticodon, and this is the primer in HIV. So even though it's a minor of these species packaged, the really interesting thing that we noticed at the time is that the ratio of lysine 1,2 to lysine 3 is similar to the ratio in cells. And this really suggested that a cellular factor that bound to all three species might be involved in the packaging process. And lysine tRNA synthetase is, to my knowledge, the only cellular factor that has that characteristic. And so we collaborated with Larry, and then years later, and then we published in 2001 this paper showing that indeed, if you look at the synthetases in the virus, you find lysine tRNA synthetase, but the other synthetases we looked at were not present in the virion. And then we went on to publish a series of other papers showing that specific tRNA lysine packaging is incorporated with the synthetase binding to the tRNA, but not amino acylation. Of course, you don't want to have an amino acylated tRNA serving as the primer. And then tryptophan tRNA synthetase is also present in RSV virions, and RSV had previously been shown to also selectively package its tRNA primer. ProRS is not packaged in the MLV, and this is a system where less tRNA pro is specifically packaged. So there's not a lot of enrichment of tRNA pro in MLV species, and the synthetase is not involved. So maybe there's a correlation between selective packaging and the need for a cellular factor that can really facilitate that. And then lysRS GAG binding, we believe, mediates, at least in part, selective tRNA packaging. So the tRNA is annealed onto the primer binding site. And so where is the primer binding site in the genome? What's shown here is the secondary structure of about half of the HIV genome, determined by Kevin Weeks and coworkers using shape probing. And the very 5' untranslated region is the site of tRNA primer annealing. So what you can see here is that the structure of the viral RNA is extremely structured. And how does the tRNA lyse 3' actually find its site for tRNA annealing? And so here's the secondary structure of the tRNA. I'm not showing the modifications here, but what we showed in my lab was that these two U's, these are the identity elements for human lysine tRNA synthetase. These are the most important critical elements for the synthetase to bind to the tRNA. And what we noticed was that upstream of the primer binding site was a pretty highly conserved stem loop sequence that had a U-rich loop here, which really mimics pretty closely the anticodon loop of the tRNA. And so we hypothesized that the synthetase might bind to this and then help direct the tRNA to the site of primer annealing. And indeed, we showed that there's high affinity binding to this site from lysine tRNA synthetase, but not other synthetases. And then if we mutate the U's to C's, the primer placement in the virion is reduced about significantly, and then viral infectivity is sort of correspondingly reduced as well. So this seems to be important for lysine tRNA synthetase to help the tRNA locate the primer binding site. And so our model for this is that, without showing you all the data, is that the lysine synthetase is involved in, of course, in binding all the tRNA lysine isoacceptors. The synthetase can bind to the what we call the tRNA-like element of the HIV genome, and then there's like an exchange that can happen where the tRNA can then be released and annealed to the primer binding site by the nucleocapsid domain of GAG. We also showed that as reverse transcriptase continues or starts initiating from the 3' hydroxyl group of the tRNA primer, the affinity of the synthetase is weakened, and eventually it falls off. So, yes, so then how is the next question? So the next question we wanted to look at is, how is lysine tRNA synthetase, if it's involved in this pathway in HIV, how is it diverted from its normal function to facilitate HIV replication? And so this is an important question because in the cell, 8 of the human synthetases, including lysine tRNA synthetase, CARS here, are bound to this multi-synthetase complex in the cytoplasm. And so this isn't a full cryo-EM structure, but it's based on structures of subdomains of this complex, and the interactions that are shown have been validated by many different methods. And so lysine tRNA synthetase, and so in addition to the synthetases, there are 3 cellular factors that are scaffold factors for this complex. And so here's where lysine tRNA synthetase is. There are 2 homodimeric copies of the synthetase, and it's bound to AIMP2 via the N-terminal 36 amino acids of this cellular factor. And so, and so now what I wanted to tell you is, why does the cell have these aminoacyl tRNA synthetase complexes? And almost all eukaryotic cells have this. The composition of them is different, but they almost all have this. And so here's just a schematic showing, again, the multi-synthetase complex. And these arrows depict other pathways that these synthetases and cellular factors have been documented to be involved in. And so we now, there's an entire field, this unanticipated biology of aminoacyl tRNA synthetases that perform non-canonical functions beyond their role in protein synthesis. And so there are Q-dependent non-translational functions. So basically, if cells are stimulated in some way, the synthetases might get modified, released from at least partially released from the complex, and then go on to do their alternative functions, which might be include being secreted, becoming nuclear factors, interacting with complexes that somehow regulate translation in other ways, and so on. And this is actually an outdated slide now, so I can probably add about 10 more pathways to this. And so the role of the MSC, we actually think, is to keep the synthetases and scaffold factors in check so that they aren't doing these promiscuous activities when they're not needed. And so what about lysine tRNA synthetase? So if you'll remember on the complex, it was pretty peripheral, and it had been shown when we started working in this area, it had been shown to be involved in at least 3 pathways. When mast cells are stimulated with IgE, the synthetase had been shown to become phosphorylated, released from the complex, and then have a nuclear function in activating transcription of certain genes. It had also been shown to be secreted upon certain stimuli, and then also in cancer cells, phosphorylated lysRS at a different site migrates to the membrane and is involved in cancer cell migration. And so we were particularly interested in whether one of these pathways might be the way that HIV recruits this cellular factor for its own purposes. And we were really interested in the nuclear function, especially after Alice Duchamp in my lab started looking at what happens to the synthetase localization upon HIV infection. And Li Wu was instrumental in helping. When Larry Kleiman retired, I had been doing a lot of the cell-based stuff with his lab, but now we wanted to get it going at Ohio State, and Li Wu was really instrumental and was part of all of this early work. And so what Alice showed is that here's the location of lysine tRNA synthetase in the absence of HIV infection, but then upon HIV infection, it becomes nuclear. And if you use a small molecule, U0126, which is a known MEK inhibitor that had been shown to prevent serine 207 phosphorylation in the mast cell system, you then prevent the nuclear localization in the HIV-infected cells. So there is a very well-known pathway for phosphorylation that phosphorylated lysine tRNA synthetase was part of. And so we proposed or hypothesized that it was the same serine 207 that is mediating release of the synthetase and its functions in HIV. Now, we had no idea what this nuclear localization was about, and I'll get to that in a minute. But first of all, is lysine tRNA synthetase actually phosphorylated during HIV infection? A few years later, we were able to get some phosphoantibodies specific to S207. These weren't the best antibodies in the world, but they showed, you could show that with, depending on the cell type, you could show that upon HIV infection, lysine tRNA synthetase did light up and suggesting that the phosphorylation event was on serine 207. So what is the role of phosphorylated lysine tRNA synthetase and nuclear entry in HIV infection? So first we made some phosphomimetics and did some in vitro experiments, and we showed that when you look at S207D, the phosphomimetic form of lysine tRNA synthetase, it binds the tRNA just fine. So there's no defect in tRNA binding. However, when you look at amino acylation, right, the capability of the synthetase to charge that tRNA, the S207D variant was completely inactive. So S207A was completely active in its amino acylation in vitro, but S207D was pretty dead. And so why is that? What is the mechanism of this inactivation? Other people did some of that work for us. So because of the interest in this phosphorylation due to previous work, this group had solved the SAC structure of wild-type LysRS and then the phosphomimetic form. And what I don't have it listed here, but the serine 207 is kind of around the linker region here between the anticodon binding domain and the catalytic domain of lysine tRNA synthetase. And so when that is phosphorylated, it really causes a major conformational change in the synthetase. And this is what's responsible for partially dislodging it from the multisynthetase complex. And so in terms of its amino acylation function, when it's not phosphorylated, it's in this, what we call now, closed conformation, where the tRNA is bound to the anticodon and the acceptor stem is in the catalytic site and is perfectly competent for amino acylation. But when the phosphorylation happens, the S207D variant is, again, opened up. The CCAN dislodges from the catalytic center, and this is now not active in tRNA charging. And so, however, what is stimulated in this phosphorylated form is another activity of amino acyl tRNA synthetases, and that is to make this dinucleotide alarmone, AP4A. And so this is a dinucleotide that can actually facilitate transcription of genes in the nucleus, and so I'll get back to that in a minute. And of course, the lack of amino acylation is beneficial for the tRNA serving, as the primer as well. Okay, so we then wanted to really see what is the effect of this phosphorylation on HIV, and we collaborated with Nate Scherer and Ryan Behrens, who were able to help us make, they made, I should say, the S207A lysRN knock-in cell line, Jurkat cell line. And so they made this homozygous knock-in, confirmed it, and showed that lysine tRNA synthetase is robustly expressed. So these cells only express S207A lysRS, so that would be incapable of this phosphorylation event. But the cells were perfectly viable because this variant is perfectly active in amino acylation. And so this was kind of our dream reagent to really try and elucidate what is the function of this phosphorylation event in the HIV life cycle. And so what we showed is that, shown here on the left is a protein expression, looking at protein expression of GAG in the wild-type Jurkat cell lines and then in the S207A cell lines. And what you can pretty clearly see here is that even though this synthetase is perfectly viable in charging and there's no effect on any other proteins, we see a significant decrease, about 75% decrease in the amount of newly synthesized GAG in these cells. We then wondered, was this an effect on the protein translation or the RNA transcription level, and showed that it was an effect on transcription. And so the transcription of the viral DNA was reduced about 75% in the S207A jerkat cell lines. So what is the role of this phosphorylation and the effect of this decrease on HIV replication? And what you can see here is a long-term spreading infection assay where here's the wild-type jerkat cells, which are perfectly happy, but here is what happens in the presence of this S207A cell line. And so pretty much these cannot support replication, and here's the beginning, just blowing up this a little bit. So they can't actually infect these cells, but there's very little replication. So this is a pretty severe defect. So the overall, without showing you all of the data again, the overall model that we have now for the function of phosphorylated lysRS in the HIV life cycle is that is shown here. So lysine tRNA synthetase is normally part of the multi-synthetase complex in human cells. When upon HIV infection, we get phosphorylation of serine 207, opening up of the structure, and so now this form cannot charge the tRNA, but it can still bind it. We showed that this AIMP2 N-terminal peptide, which is the peptide that keeps the synthetase in the closed conformation in the complex, actually will prevent these downstream events from happening because it prevents this form. If we overexpress AMP2 in the cell, we prevent this from forming, and it really keeps the synthetase in the closed complex. Now, there's two functions for this form, the phosphorylated form. It can interact with GAG, and that can result in promoting tRNA assembly and packaging. And it also goes in the nucleus. And the nuclear form has enhanced synthesis of AP4A. This dialarmone binds to HINT and then releases USF2, which is a transcription factor which binds up the promoter element in HIV and promotes HIV proviral DNA synthesis. And again, I didn't show you all the data for that, but this is the nuclear effect that we think is happening. And I just want to advertise a poster by Mafam in my lab, and she is investigating a third possible function. These are all positive functions for HIV life cycle in suppressing the CGAS sting immune response. And so she has a poster tomorrow, 2/12, and so please take a look at that. And I think this is starting to be a possible third function for lysRS in the HIV life cycle. Okay, so in the last 5 or so minutes, I'm going to talk about the other function of GAG besides the chaperone activity that we've been really focusing on in the last few years especially, and that is its function in genomic RNA packaging. So 95% of the HIV virions package 2 copies of the viral genome as a dimer. But the binding preference, if you just study siRNA binding versus some non-siRNA binding in vitro, is not very strong. So the discrimination at the binding stage is maybe less than tenfold. In infected cells, the ratio of genomic RNA to messenger RNA is probably less than 1 in 10,000. Be happy to hear what your favorite number is, but this is around what we're saying. And so how does GAG find this needle in a haystack? So in recent years, it's become clear that when viral RNA is synthesized by RNA Pol2, it really has heterogeneous transcription start sites. And so whereas the proviral DNA encodes 3G at the start site, you actually get heterogeneous mix of 3G, 2G, and 1G. And so the work that I'm going to tell you about is in collaboration with Wei-Su Hu, and what Wei-Su's lab showed is that if you look at the viral RNA in the cell, it's mostly 3G, as you would expect. But if you look in the virus, the preference for packaging the viral genomic RNA is for the 1G species. And so the 1G RNAs are preferably packaged as genomic RNA, whereas the 3G RNAs are preferentially translated as messenger RNA. And I'm not going to show you that work, but again, in collaboration with Wei Su's lab, and then also Telznitzky, Scherer, and Summers published a paper around the same time showing that the 3G RNAs, that both RNAs can be translated, but 3G RNAs are preferentially translated. So but the question is, what is the mechanism by which a difference in two guanosines at the very 5' end of the genome dictates this unspliced full-length viral RNA fate? And so we carried out structure probing studies to test the hypothesis, well, maybe the structures of these RNAs are different. And this is data from Joe Canlong in my lab, and sure enough, what he showed was that in the case of the 1G RNA, we have a structure where you have an intact TAR, intact poly A, you have an exposed dimerization initiation sites and exposed GAG binding sites. Whereas if you look at the structure, and this is all using shape map probing, if you look at the structure of the 3G RNAs, where we're just adding 2 more Gs at the very end here, what happens is that next G steals this C over here and totally disassembles the poly A hairpin. And so now you have a poly A hairpin and SL1 that are completely refolded, the DIS is sequestered, and the GAG binding sites are distal and partially exposed. And again, about the time we were working on this, some other labs were working on this too, and although the details are a little different, the basic agreement is that the TAR poly A is intact here and it's misfolded here, resulting in these other downstream changes. So our model is that the 5' UTR can adopt different conformations depending on the number of Gs. The 1G RNA favors this conformation with an intact poly A hairpin. This results in an exposed DIS, so this can dimerize, and exposed GAG binding sites, and this RNA is packaged as a genome. In the case of the 3G RNA, it forms a structure where the poly A hairpin is not folded, and in its normal conformation. The DIS dimerization site is sequestered, and the GAG binding sites are also sequestered, and so this is not packaged as a genome. So this really tells us something about the poly A hairpin stability, that there's really a major difference here that is really resulting in a lot of these downstream effects. So to look at that a little more closely, what we wanted to do is actually measure the melting temperature of these RNAs. And so here again is the wild type system showing that the 1G is preferentially packaged and the 3G is not. And then, so what happens to actually the stability of the TAR poly A region? So if we look at the RNA structure prediction program, it will tell you that they have exactly the same stability. But if you actually go to measure the melting temperatures of these hairpins, they melt in two stages. So you have an initial melting profile, that's for poly A, and then you have TAR melting. And what you can clearly see if you compare the 1G and the 3G RNAs is that the TARs are fairly similar, but the poly A's, there's a 9 or 10 degree difference between the melting of the poly A hairpin, with the 3G being much less stable than the 1G. So this really shows that there's a significant difference in the poly A hairpin stability, but how does that really impact, packaging? So what we did was make Jonathan Kitzrow in Wei Su's lab made some transcription start site mutants. So instead of having a viral RNA that started with GGG, he mutated that to GGA. That would mean the virus has the chance to package either a GGA or a 1A in this case, because this would be equivalent to where the 1G normally is. And sure enough, both species are packaged. The GGA and the 1A are transcribed and packaged, but there's no preference. So with this mutation, there's no preferential packaging of the 1A RNA. So what happens to the poly A hairpin stability? So what we see is that, again, there's a small difference in the TAR stability, but now the stability of the poly A hairpins is identical. And so the equal stability of the poly A hairpins eliminates, is correlated to the elimination of selected packaging, of these RNAs. So then Jonathan designed another mutation, and this, and the structures, I'll just show you real quickly. The structures of these are now identical too. So we now have these differences in the structures of the 1A and the GGA RNAs. So he designed a mutation where now we have an additional mutation in TAR, And can't go into details, but we predicted that this might affect the different structures of the poly A and the different stabilities. And what we see with this mutation, which has the GGA transcription start site, but this additional mutation in TAR, what we see is now this now selectively packages the 1A. So we've restored the ability to selectively package the 1A RNA. And so what about the poly A hairpin stabilities? In these constructs, we now restore the differences in the poly A hairpin stabilities as well. And as you might imagine, the structures are also now significantly different, again, where you have an intact poly A hairpin in the 1A, but not in the GGA. And so together, this work suggests that transcription start site mutations that abolish poly A hairpin stability differences between the 1G and 3G RNAs abolish the selective packaging. Okay, so maybe I'll just tell you one more short thing here, and that is that we predicted from this RNA that we would also have preferred GAG binding to the 1G, because this has the exposed GAG binding sites and the DIS is also exposed. So that's the prediction. But it turns out that no matter what type of in vitro binding assay we do using standard physiological conditions, it's very difficult to see significant differences in the binding of GAG between the 1G and the 3G RNAs. But what we decided to use was use mass photometry, and this is a solution-based mass technique which allowed us to look at the stoichiometry of binding of GAG to the 1G 5' UTR or the 3G 5' UTR. And this was Kaylee and Vicky Wysocki's lab at OSU. And what we showed was that the blue is the 1G RNA, that on the 1G RNA we see a buildup of 1 GAG, 2 GAGs, 3, 4, 5, 6, and higher order GAGs, whereas under the same conditions with the 3G RNA, we only see a single smaller complex forming. So clearly there's really a difference in the binding of these RNAs and the assembly of the GAG on the 1G RNA. Now, a few years ago, we showed that when GAG binds to Psi, the effective charge of that binding is 3 to 5, whereas binding to some non-Psi RNA, the effective charge is about 3 times higher. And we hypothesized that maybe when it's binding to Psi, it's using only the NC domain, versus when it's binding to some other RNA, it's binding in kind of a bent conformation with the other nucleic acid binding domain also contacting the RNA, resulting in this higher effective charge. And so to test this hypothesis, Yahong in my lab has developed, again, another FRET-based approach to look at the conformational changes of GAG when it's bound to different RNAs. And I'm not going to steal her thunder because she's actually giving a talk on Friday. But just to give you a little preview is that that's exactly what we see when we have this start out with a high FRET bent conformation of GAG, and we add TAR poly A. We've pre-incubated the GAG with tRNA here because that's its natural state in the cell. We add TAR poly A, and we really don't see much of a conformational change. But when we add Psi RNA, this GAG undergoes a significant conformational change to a more extended conformation, suggesting that it's really promoting GAG-GAG multimerization on Psi, but not on the non-Psi RNAs. So I really encourage you to go to this talk. I think it's a very exciting finding, and she's also going to talk about the role of IP6 in this discrimination. Okay, so I've just told you everything I think I have time to tell you today, and so this is our current model for specific genomic RNA packaging, that transcription start site differences determine the RNA fate, that 1G RNA is preferentially packaged due to favorable structural features, that poly A hairpin stability differences are really critical for this selective packaging, and that GAG assembles more readily on 1G RNA relative to 3G RNA. And then this conformational change of GAG from the compact to the extended form is really promoted very efficiently by Psi binding, but not by other RNAs binding, and that the binding of cellular factors tRNA and IP6 also helps GAG to distinguish between Psi and non-Psi RNAs. So I just want to end by thanking the many collaborators, many of whom are in this room, for all the very fun collaborations over the years, for teaching me about retroviruses, And I hope that we can continue to collaborate for many years to come. I also need to thank my group. What really keeps me moving forward, of course, is my trainees and people that I can work with every day in the lab. And here I'm just listing my current group. And again, Mafam and Yehong have posters here, but they're great. And over the years, I've had so many great trainees, many of whom have also attended Cold Spring Harbor. And of course, my family, who keeps me grounded in non-science things. And I want to end by encouraging you all to attend another conference, and this is the International Retroviral Symposium, which is going to be held in Prague this fall. So please register for this. It's the deadline is June 10th, I believe, for registration. So thank you.
Questions for Karin? I could ask you one.
Oh, there's one over there, I think.
Hey, fascinating talk, especially the RNA packaging part. I remember maybe reading a paper about proviruses that are deleted in potentially the packaging signal, and them still being able to produce virions that package RNA. So I was wondering if you had any, if that would be an area to investigate, if there are proviruses with certain deletions that still retain maybe that stem loop that you were talking about, so they could still package RNA potentially.
And was that specifically in HIV?
Yes, yeah,
Oh,
HIV. So yeah, I guess my curiosity was, is it possible that the packaging signal could be deleted and these proviruses still make RNA that's then packaged? Or is it maybe other RNA that
Definitely other RNAs are packaged. We know that if the genomic RNA is not expressed, if you just express GAG, it will package cellular RNA, and the virion will still assemble, but it won't be infectious.
Got it,
in that
okay.
case, yeah.
I guess you've collaborated with the rest of the meeting.
I think there's one up there.
Hi. Sorry for a non-retrovirology question, but this has bugged me for a while. I wonder if my understanding is that the class 1 and class 2 amino acyl tRNA synthetases have like no structural homology, right? Like they're very different enzymes.
They have different, different. Well, they perform the same reaction,
Right,
but
right.
they have two different
But
distinct
their
catalytic
structure, yeah,
sites.
yeah.
Yes.
Do you have any thoughts on why that is, how that could have evolved, and why?
Oh, that's a beer question, I think. So yeah, it's very interesting that if other people don't know, there are two classes of amino acyl tRNA synthetases, and they have evolved independently. And there's a lot of discussion in the field in terms of how that actually happened, and it really is probably too much to answer right here. But it's they're a fascinating family of enzymes because if you think about it, they're the first enzymes, among the first enzymes anyway, to have had to evolve. So, yeah, it's a great question, though. Oh, yeah.
So how important are the tRNA modifications for your structure that you just had?
They're critical for proper, for example, docking of that N-terminal alpha helix. So nobody had ever seen that N-terminal alpha helix even form until we made the structure with the modified tRNAs. And what happens is that there's a dihydrouridines in the tRNAs that normally if they're used, they stack on each other. And when they're dihydrouridines, they're so flexible that N-terminal helix of LysRS can actually have an induced fit, interaction with that region and actually form, interact with the groove of the tRNA. So I think they're critical for that. We know that they're really important for binding affinity and I think for selection of the correct tRNA, in cells. In terms of HIV, we really want to know more about how their function in tRNA annealing and in interacting with, different RTIC. So we're trying to continue to collaborate with Yang Zhang on forming the structure of the RTIC with the modified tRNA. Yep.
Okay, I see no.
Oh, let me see.
Karin, thank you for the great talk. I have a more personal question to ask you. For all the time I know you, you have been very positive about everything, and started from like talk you gave today about inspiring us. So I want to know how you, even in time that doesn't look so good, you can keep being so positive.
Well, I think, as I tried to say at the end, I think I have to be positive for the people around me, I think. And I'm just continually inspired by the students and the trainees in my lab, and I know that I want them to succeed in science, and so I think we just have to keep going and do the best we can, even though the times are a little tight right now, but to do the best we can with the resources that we have. And continue to collaborate, organize meetings. We had this, discussion, should we cancel this meeting? Are people going to come, But we decided to move forward, and we hope that many, many of you do end up attending. So. Oh, Johnson.
So, Karin, thank you for the talk. I should know this, but I'm wondering, among all these modifications in tRNA, like there are a lot of different type of modifications, how many of these have been tried and applied to mRNA vaccine to see the stability, delivery effect, like in the practical, well, the translational sense?
Yeah. That's a very, very active area of research, and obviously in the vaccines that were developed, everybody probably knows it's a they're pseudouridine, methylated pseudouridine that really ended up being the key for avoiding the immune response. But that's a very active, I don't know how many off the top of my head, but that's a very active area of research, and people are trying to apply these modifications to make even more stable and more highly produced, mRNAs.
Yeah, because I think at the moment they could have modified the genome to bring it down to uridine level down to roughly 12%. So I'm just wondering, any insight? Is it what other modification did it have? Did it have much on the AGC residue, or just mainly in U?
There are modifications on I think every single residue. U is one of the most highly modified, but yeah, I mean I don't have a lot of insights into all the details in that area.
Okay, I see no more hands. All right, thank you, Karin. That was great. Very great.